Welcome to AP Biology. We have talked about signal perception already and different intercellular transduction pathways, and now we're going to talk about the cellular response or different possible cellular responses. The cell response can be kind of anything. So, let's look at some examples. So let's say a growth factor or some ligand, right? In this case, let's say a growth factor attaches to a receptor. This is going to be very specific. It's going to change the shape of the cytoplasmic domain of the receptor, which is then maybe going to activate a protein, which activates another. Maybe it's each in turn phosphorylating the next. So we've activated a bunch of proteins. Finally, this one comes into the nucleus and takes an inactive transcription factor. So what does transcription mean? This is a unit we haven't done yet. You know how DNA makes RNA, and RNA, if it's messenger RNA, will make a protein. So transcription is this part. The DNA makes the RNA. So here's an inactive transcription factor. This growth factor has led to a cascade that turns on an enzyme that activates this transcription factor. It phosphorylates it, and now it's able to go to one particular gene in the nucleus, one particular piece of DNA, and act as a transcription factor. Say, hey, we need to make RNA of this type. So we saw before how testosterone can do that when it goes right through the lipid bilayer. Now we have an example of a water-soluble or hydrophilic growth factor instead that goes to a receptor on the surface and ends up causing the same thing, a transcription factor. So one result of a signal transduction pathway can be to activate a particular gene. Say, hey, we need some RNA and then we're going to need some protein. Another case is where you already have all of the enzymes that you need in order to do what you need to do. So let's say epinephrine binds to a receptor and then it triggers a whole cascade of reactions. Well, what really needs to happen is not that we need to make this enzyme, but we need to activate it. So this enzyme, glycogen phosphorylase, is an enzyme that breaks down glycogen to make glucose. And it's already here. It's already been transcribed and translated. It's already ready to go. It just needs to be activated, and this signaling molecule will activate it. So if you need a response really, really quickly, your cells will usually already have the enzyme made, but it will be inactive and then it just has to get activated. That's pretty quick. If the cell can maybe doesn't want to waste the resources on something that doesn't happen a lot and it doesn't need to happen quickly, then instead it will take a little bit longer to actually turn on the gene. So you can do these two different things, either turn on a gene or already have the product from that gene produced, but in an inactive form, and it just needs to be activated. Another example is to change the behavior of a cell, for example, cell shape. And so we looked at this one before where a mating factor caused, I think it's a yeast cell, to activate a G protein, which then activates this thing called fuse3, which then activates this thing called formin. And then it makes the cell grow in a particular direction by assembling actin subunits. And so it's making these schmooze to grow in a certain direction. And so that's just another example of a response, the growth in a particular direction. And so AP might give you this and ask, hey, what happens if the ligand binding site is mutated? The DNA that makes this receptor maybe has a mutation in this part, or maybe instead it has a mutation in this part, so it's always on. In that case, you wouldn't need the mating factor at all, it would just always be on. Or what happens if there's a mutation in the fuse3, et cetera, et cetera. So there's lots of things they could ask here. Okay, next idea, the specificity of cell signaling and coordination of the response really depends on the kinds of receptors that you have. So here's a target cell for hormone A, and it does not fit. It doesn't have a receptor for hormone B, which is the triangle one. And this one is, it has a receptor for hormone B right here, but it doesn't have a receptor for hormone A. So this is just the idea that different proteins allow cells to detect and respond to different signals. So this one can respond to both. This one can only respond to hormone A, and this one can only respond to hormone B. So not every cell responds to every ligand. Okay, this is the idea that the same ligand causes a different response in some cells compared to others. So for example, insulin is going to be these green arrows. And so insulin will cause glucose to be taken up by the liver cell, the muscle cell, and the fat cell. But in the liver cell, the glucose will also be converted to glycogen, whereas that might not happen in some of the other cell types. And then also, like if we're looking at if we're looking instead at glucagon, a different ligand, right, a different hormone, it will cause, let's see, the liver cell to break down or to release glucose. Um, and it will cause the liver cell to take up amino acids, but it's not going to cause muscle cells to take up amino acids, for example. Okay, pathway branching crosstalk further helps the signal to coordinate the signal coming in and coordinate different signals. So, for example, right here, let's say you have insulin attaching to a receptor and it's causing you to take in glucose. Later, you might have glucagon released and it goes to its receptor and it can cause the inhibition of that response so that maybe you're not taking in glucose so much. So those two can really work against each other. This one can inhibit the other one. So this is just another way to help coordinate your cellular response. Okay, next idea, fun fact here, scaffolding proteins. When you think of a scaffold, maybe you think of someone up on a tall building washing the windows, or maybe you think of a construction worker working on the roof of a building and they need a scaffold to stand on. So a scaffold sort of helps them to work, maybe puts all their stuff in one place so you can have maybe some of your tools up there with you. So in the cell, a scaffolding protein will be a protein that holds different parts, different relay proteins, different parts of the signal transduction pathway together. So instead of one protein activating another, which then has to diffuse around to find its its reactant, and then it causes that to become a product, and that maybe in turn is going to diffuse somewhere and then find its reactant to turn that into a product. Instead, if you have them all in the same place, the ligand binds here to the receptor binds to the scaffolding protein, which maybe causes a change here. And then this one, once it's changed, doesn't have to diffuse. It's sitting right next to the thing that it works on, its substrate. Which then works on, once it's activated, works on the next one. So the scaffolding protein is going to help to increase the signal transduction efficiently by grouping these parts of the relay together. Okay, finally, you are going to need to terminate a signal. So if you start a signal transduction pathway, at some point you have to turn it off. So sort of like anything in your life, you turn on your computer, eventually you should probably turn it off, right? You're playing on your phone, at some point it's time to put the phone away, believe it or not, right? So for example, if your nerve cell in your arm sends acetylcholine, a ligand, to your muscle cell to say, hey, it's time to contract, and that causes a bunch of it causes a particular signal transduction pathway in the cell, which causes your muscle to contract, right? You can't have your muscle contracted all the time. You've got to relax it and like pretty quickly, right? So that acetylcholine is always bouncing on and off of its receptor. There's an enzyme, acetylcholinesterase, which will actually break down your acetylcholine. So here's your end of your nerve cell, which has the acetylcholine in vesicles. That acetylcholine will be released when the impulse goes along the neuron. Here's your muscle cell. Here are some receptors for acetylcholine. That will then trigger the whole muscle cell to be stimulated if there's enough of them, which causes sodium to go in, which causes calcium to be released inside from your endoplasmic reticulum, blah, 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 causes it to contract, right? But there's going to be an enzyme sitting right here as well called acetylcholinesterase. And when this ligand bounces off and bounces over to it, it will break down the acetylcholine so that your muscle will be able to then relax, right? You can't have it constantly contracted. That would be a bad thing. So unbound receptors revert to the inactive state, and even the pathway inside, you're going to be able to inactivate that as well. If you phosphorylated some enzymes, you're going to you're going to have other enzymes that can dephosphorylate or take the phosphate off. Okay, this is the idea that the environment can affect the cellular response. So for example, as soon as this antelope starts getting chased, it's going to be releasing epinephrine. In fact, it'll probably start releasing it as soon as it glimpses the cheetah, right? And the epinephrine is going to cause it to, among other things, break down glycogen so it has more energy for its run. So it has more glucose, and then the mitochondria can take the glucose in, break it down, make a lot of ATP, and that'll be necessary to get away from the cheetah. So that's an environment, right? That's the cheetah. As soon as the antelope or whatever this is sees the cheetah, it's going to be able to release the epinephrine. So that's the environment telling the cells what to do. It could be the internal environment instead of the external environment. So it could be that you ate a bagel this morning, and that bagel was broken down to the starch in it was broken down to glucose, released into your blood glucose levels are supposed to be here, but they went up because of that bagel. And now you release insulin, which is going to tell your cells to take in glucose and bring the glucose levels back down. And so that's an internal environment triggering you to release insulin, which then tells your cells to do something and causes some kind of cellular response, like taking glucose in. So it can either be the internal environment or the external environment that tells your cells what ligands to release.